medicine. Our independent scientific advisors choose from scientific proposals submitted from all over the world to make sure that we fund studies with the greatest merit and the highest impact for animals. This next slide shows a chart that gives a breakdown of how we currently allocate our research dollars. As you can see, most of the research money does go to companion animals such as dogs, cats, and horses. However, almost a quarter of our dollars go to fund wildlife species such as bats. Most people don't realize that Morris Animal Foundation is by far the largest funding source for research to benefit the health of wildlife species. Each year, we fund at least $1 million of research just for the benefit of wildlife. In fact, we've funded research that has benefited more species in more places than any other nonprofit organization. And when veterinary researchers need help responding quickly to unexpected events, Morris Animal Foundation answers the call through our Betty White Rapid Response Fund. As many of you may recall, earlier this year, many of those cute little guys you see in the picture on the right were in trouble, with more than 1,200 of them dying off the shores of California. With the help of our Rapid Response Fund, researchers from the National Marine Mammal Foundation were able to quickly step in and work to identify the costs of the loss of these cute little baby California sea lions. This chart shows a breakdown of how we spend our wildlife dollars. Although general health, nutrition, reproduction, and various other medical topics are addressed by our funding, the vast majority of our funding goes to solve problems of the greatest need in wildlife, infectious diseases. And today's topic, white nose syndrome in bats, is no exception. At this time, I'd like to introduce our speakers and experts in white nose syndrome research. Dr. Michael Sorensen is a professor and chair of biology at Boston University. His research not only includes work with bats, but also many species of birds, fish, and ants. His areas of interest include avian behavioral ecology, speciation, population genomics, and molecular systematics. Ooh, that's a mouthful. Dr. Sorensen is joined today by Erin Wilder. Erin is working on her PhD in biology at Boston University, and she's performed a lot of the research in both the field and the laboratory on the project that we'll be talking about today. Dr. Sorensen would also like to acknowledge the work of Dr. Tom Coons, one of the world's leading bat biologists, as a contributor to this research. At this time, I'd like to turn the presentation over to Dr. Sorensen and Erin so that we can learn more about our topic, white nose syndrome in bats.
Hello, everybody. This is Mike Sorensen.
And this is Erin Wilder. So I'm going to start off and tell you a few things about bats that you may not have known. Bats are in the order Chiroptera, so they're not closely related to birds or rodents. Chiropterans are a very diverse order. There are over 12,000 species of bats. Bats are the only mammals capable of flight. And they are nocturnal, so they use echolocation to navigate at night. And contrary to popular belief, they don't actually fly into people's hair, at least not on purpose. Next slide, please. There are 25 species of hibernating bats in North America. 4 of those species are endangered and 13 are of conservation concern. Bats are considered true hibernators, which means that they drop their body temperatures down to near the temperature of the cave or mine that they're hibernating in for long periods of time during the winter. So this figure on the right here is a thermal image where the cool colors represent cool temperatures and the warm colors represent warm temperatures. So what we're looking at here is a cluster of bats hanging from the cave wall. And you can see that the cave wall is green because it's a cool temperature, and most of the bats in this cluster are actually close to the cool temperature of the wall, except for the little bat on the left, bat number 2, which is warm. That bat has warmed its body temperature up to normal mammalian temperatures, which they do periodically during hibernation. As I mentioned, they hibernate in caves and mines, and these winter hibernating bat colonies can reach hundreds of thousands of individuals in the northeastern United States. Next slide, please. So it's during this sensitive hibernation period that white nose syndrome strikes. white nose syndrome was discovered in 2006 in Howe's Cave, which is a cave near Albany, New York. It's caused by a cold-growing fungus shown here on the right. It's cold-growing, which means that its optimal growth temperatures are pretty much in the range of the temperature of a hibernating bat. And because hibernating bats also have suppressed immunity, they actually represent a really susceptible host to this fungus. So this bat here on the right has the characteristic fungal growth patterns for which white nose syndrome was named, growth on the muzzle, as well as growth on the wings. And the fungal growth on the wings can actually cause quite severe wing damage. The picture on the bottom left shows a bat that's missing a good chunk of its wing membrane as a result of fungal growth. And the wing damage is actually thought to play an important role in the cause of death from white nose syndrome. Next slide, please. So mortality from white nose syndrome in hibernating bat colonies approaches 100% over several years. So this figure on the right shows state census data of the hibernating winter colonies in the Northeast. And so you can see since the 80s, most of these hibernating colonies were growing steadily or were stable until white nose struck in 2006. Most of these colonies at that time plummeted to near zero. There are 10 hibernating bat species affected by white nose syndrome. And as of 2012, an estimated 5.7 to 6.7 million bats have died from white nose. Probably more have died since. The regional extinction of at least 2 species has been predicted, the little brown myotis on the bottom left and the Indiana myotis on the bottom right. Next, please. So following the emergence of white nose syndrome, it spread rapidly throughout eastern North America. So in this map on the right, there's a red circle surrounding Howe's Cave right there in the center of New York. And the counties here are highlighted according to the year that that county reported white nose syndrome. So you can see just how quickly it spread throughout the east. And a critical component of our understanding of white nose syndrome and how best to respond to it is understanding its geographic spread. Understanding the patterns of spread will help us to predict future routes of spread and identify bat populations that are most at risk of white nose syndrome. And this information will help us to better target disease surveillance and mitigation strategies. So the first thing we did was look at white nose syndrome risk factors to hibernating bat colonies. And we looked at a data set that had been collected by state agencies in the Northeast for the last 30 years. So we looked at characteristics of winter colonies and how that relates to their risk of white nose syndrome. What we found is that the risk of white nose syndrome increases with colony size. So the bigger the hibernating colony, the greater the risk of white nose syndrome. It also increases with the proportion of the little brown myotis species that's hibernating in that colony and the proportion of bat species that tend to hibernate in humid portions of the cave. Next slide, please. So another critical component of understanding how bats move across the landscape is, or understanding how bats spread white nose, is understanding how they move across the landscape. So here I've shown 3 hypothetical populations. The red population represents one infected by white nose syndrome, and the green and blue represent susceptible populations that don't yet have white nose syndrome. So you can imagine if you have more bats moving between the red population and the blue population, you'll have more bats moving the white nose fungus to the blue population than to the green population. And you would expect white nose syndrome to hit the blue population first. Next slide, please. So bats also move their DNA between populations. Oh, back one slide, please. Bats also move their DNA between populations, so you would expect that if more bats are moving between the red and the blue population, those populations will look more genetically similar than the red and the green populations. And we can use this information to tell us how bats move between populations and then how the fungus might be introduced to new populations. So we look to the little brown myotis to help us understand how bats are moving across the landscape. So the little brown myotis is this handsome guy in the bottom right-hand corner. We chose this species because it is a highly white-nose affected species. It is common and wide-ranging, so you can see in the range map on the right, it ranges throughout much of North America, and it's common through much of that range. It's also one of the species whose regional extinction from the Northeast was predicted within the next 20 years based on the mortality rates we saw initially. Next slide, please. We sampled little brown myotis throughout their range. These red dots represent sites from which we have tissue samples, either collected by us in the field or collected by other researchers already catching bats in the field for other research. Next slide. So there are a number of ways to catch bats in the field. Most commonly we use mist nets. And mist nets are a fine mesh net that bats have a hard time detecting using echolocation. So we stretch these mist nets across rivers or streams or other places we expect bats to be flying through. And bats will fly into the net and become entangled. So we'll remove, once we have a bat in the net, we remove the bat and this it right away. We'll hold it in these soft cloth bags individually, and the cloth bags just sort of help it feel less stressed and a little more cozy. Once we're ready to process the bat, we take it out of the bag, we identify it to species and take some baseline measurements. We measure the forearm and the weight of the bat. We look at the sex and reproductive status, and we also look for signs of wing scarring that may indicate white nose and its severity in that population. And finally, we take a small tissue sample from the bat's wings. So you can see in the bottom right picture, I've circled the little wing biopsy site. And it's from this wing biopsy that we can extract the DNA. So once we're done processing the bat and we have our genetic sample, we let the bat go, and the wing punch heals within 2 weeks with no detectable negative effects on the bat. Next slide, please.
So I'm going to jump in here and talk a little bit about the genetic analyses. So from those little bits of wing tissue, we extract DNA from the bats. And we use that material to look at genetic variation in the bat population. And one of the things that's very exciting about this work is that we're now able to use the same sort of advanced methodology that's been used in human population genetics these days. So something called next generation DNA sequencing, which allows us to survey genetic variation across a large part of the bat's genome. Some of you may be familiar with National Geographic Society's Genographica project, in which you can actually send in a sample of your own DNA and get back very detailed information about your ancestry and where your family came from geographically. And we're using basically the same approach with the little brown bats to look at the geographic structure of their population. Next slide. So here's a somewhat complicated diagram, but I'm going to try to explain the basics of it to you. This is a summary of thousands of individual data points from each bat, thousands of pieces of information about genetic variation, all sort of distilled down into one simple figure. And so in this diagram, each individual colored dot represents an individual bat. And then the different colors and the ellipses surrounding the points of a given color represent different geographic locations. And in this case, each of those colors represents a different state or a different Canadian province. In general, the way to think about this diagram is that 2 points that are very close to each other in this space are genetically similar, whereas points that are further apart are genetically more different from each other. And as Aaron is going to show you here in the next few slides, one of the remarkable results is that this plot of genetic variation turns out to mirror the geography of the populations in a remarkable way. Next slide.
Okay, so I'm going to focus in on 3 clusters in this figure. The cluster on the right, which represents eastern bats, the cluster on the left, which represents western bats, and the cluster in the middle, which is bats in central North America that span the 2 populations. Next slide. Okay, so zooming in onto that eastern cluster, in the map on the left, I've plotted the capture locations of the bats that are shown on the right. So we can see here there's lots and lots of overlap of these different ellipses. So lots and lots of genetic similarity of bats sampled as distantly as New Brunswick, that's NB on the map, and North Dakota, ND on the map. So this represents a really large geographic area that's very genetically similar. So this tells us, this indicates that bats are moving very widely, and bat populations in this region are very highly connected. And this is consistent with what we've seen with the rapid spread of white nose syndrome, just high connection between different populations and potentially lots of fungus moving through these different populations. Next slide. So in the West, we see a different picture. We see populations with very little overlap, and the genetics of populations, as Mike was saying, roughly mirrors that of their geographic capture locations. And this indicates that as two sites that are more geographically distant are more genetically dissimilar. And the contrast here between the east and the west suggests that bats are moving far and far smaller distances compared to bats in the east. We also have Montana, Saskatchewan, and Alberta spanning the space between the east and the west. And this suggests that bats in this region have DNA from both the east and the west, and that there is some connection and western populations. The higher geographic isolation of bats in the west could be good news for bats. If white nose does get this far, we would expect slower spread among these different populations. Next slide. So I've grouped bats here by the state or the province from which they were captured, and those are political boundaries that aren't particularly important to a bat. So you can see Montana here stretched across a fair bit of geographic space. In Montana, we actually sampled 2 sites, and so if we, instead of grouping all Montana bats together, if we split them into those 2 Montana sites, next slide please. One more. We actually see that the westernmost site, GNMT, which is Glacier National Park in Montana, falls clearly into the west, whereas MFMT, which is McClellan Ferry in Montana, falls clearly into the east. And although these sites are separated by only about 200 miles, there's clear division between bats east and west of this population. So that tells us that this separation represents a big barrier to the flow of DNA and likely represents a temporary barrier to white nose syndrome. So overall, our genetic findings suggest that spread of white nose may slow as it moves across the Great Plains and approaches this east-west barrier, and will be slower in general in the west compared to the east. Next slide, please. So just to sum up, our genetic data tells us that eastern populations are connected by very frequent dispersal, which is consistent with what we've seen with the rapid spread of white nose syndrome. Based on census data, we would expect that white nose will move more slowly in the west because hibernating bat colonies are smaller. Also, based on our genetic data, we find that populations in the west have more geographic isolation, which is also associated with slower spread of white nose syndrome. So there are currently 10 white nose affected species, and I've shown them here on the right. The species highlighted in red were already endangered before white nose syndrome struck. And there are 15 more hibernating species in North America that may be vulnerable to white nose syndrome. So it's important to realize that this study is not only aiming to increase our understanding of white nose in little brown myotis, but will also be applicable to all of these white nose affected species and those vulnerable to white nose. Because barriers to bat dispersal are likely to be shared among species, we expect that our data will be highly applicable to all affected bat species and that our response would be the same for different bat species. So I'd like to thank Morris Animal Foundation for funding this study. Next slide, please. And I'd like to thank you for listening. Happy Halloween, and if you have questions, we're all ears.
Thank you so much. We do have time for a couple of questions. Winona will start off with the first question that was asked by someone on the line.
So this question is, because a decrease in bat populations can affect crop and food production in the U.S. due to increases in harmful insect populations, has there been any financial support of critical bat research by the U.S. government?
Yes, I think the United States Fish and Wildlife Service has been very interested in the white nose problem right from the outset, and they provided significant funding for a wide range of white nose related research. The National Science Foundation as well has provided some pretty significant funding into different aspects of the disease. Our project is tackling one particular question about the genetic structure of little brown myotis populations. We're actually hoping to move into some additional analyses of immune system genes in these bats. But there are a very wide range of questions, some of it involving the social behavior of the animals and how the disease is actually spread, questions involving the healing from the wing damage that occurs during the winter time. These bats actually can heal very quickly once the weather warms up in the spring, and there's interesting questions about how well those bats do in the long term. And so there's really a broad range of questions, and the short answer is yes, the different government agencies have invested significantly in this.
Thank you. We have another question here. Are there any theories as to why western bats don't seem to travel between populations as much as those in the east?
Yeah, so there are definitely fewer hibernacula. So hibernacula are caves or mines where the bats actually hibernate. So if you actually look at a map of cave and mine entrances, the east is very densely populated by these caves and mines. There's a karst formation that really sets up very good habitat for bats to hibernate in. So, so that's one reason probably that there are fewer, there's just fewer hibernating habitat there. But yeah, there are a number of reasons, and we don't know exactly why that's the case.
It's actually the pattern we've seen for little brown myotis in the West is actually seen in a lot of different species if you compare eastern and western North America. And the West has got mountain ranges and deserts, and there's just much more topographic and geographic diversity in the West, which probably serves to divide populations of different areas.
And one last question. We've heard that cavers, or people who explore caves, might have caused or contributed to the white nose syndrome. Can you address that?
Yeah, so it's highly likely that humans are responsible for introducing the fungus initially. So it looks like the fungus actually came from Europe originally. And it was probably brought over by a caver or someone touring the cave. Howes Cave, the first site that white nose emerged in, is a big tourist site. It's actually a commercial cave where people can pay money and go in and visit. So it's very likely that the first introduction was human. And after that, there may have been some instances of humans moving the fungus around, but we also see white nose in caves that have been long gated, where gates are put over the entrance to the cave or mine, so that humans can't actually access it. And we were seeing white nose in those caves as well. So by and large, bats are responsible for spreading it, but it's possible that there are instances of humans moving the fungus around and potentially introducing it to new areas, but we don't really know. But U.S. Fish and Wildlife and other state agencies have taken a lot of measures to reduce the chance of human introduction. So there are decontamination protocols. Every time a caver goes into a cave, they're supposed to bleach their gear and disinfect before they visit another site. And same with researchers. Anytime we go into a cave or capture bats, we have decontamination protocols to minimize human introduction of the fungus.
Great. Thank you so much. That's all the time we do have for today on this Halloween. We do have a few questions at the conclusion of the webinar that we would ask for you to take a few minutes to answer. Your feedback will help us continue to bring relevant topics to you for future webinars. Thank you again, and happy Halloween.